Bismillah ar-Rahman ar-Rahim. Assalamu alaikum. This is me, Atiq Ahmad, and today we are going to start a new and fresh unit, unit number 4, that is cells and tissues. Today we will study the first topic of this unit, and that is microscopy and light microscopy. Now we'll move towards the introduction. What is microscopy? Basically, microscopy is the use of microscope. The use of microscope is called microscopy. Now, microscopy basically opened new avenues for the scientific research. Due to the invention of microscope, the research increased at a very rapid pace in the field of biology, physics, chemistry, etc. Basically, in biology, the invention of microscope basically the result resulted in the discovery of cell and its organelles. Before microscopy, there was no idea that how the living organisms are made, from which item, from which things they are composed. This is due to the invention of microscope, which resulted the biologist to observe the cells, and then later on, due to the advancement of microscopy and the invention of electron microscopy, then there are two types, transmission electron microscope and scanning electron microscope. So these electron microscope then enable the scientist to observe the inside organelles of the cells. So microscopy basically brought a revolution in the field of biology as well as in the other field, in the field of physics, in the field of chemistry, etc. The first microscope was basically developed by Zacharias Janssen. He was basically a glass maker. He has an optic shop, and this was accidentally discovered by Zacharias Janssen and his son. They basically placed two lenses in a tube and observe small things with that tube. So when they observed that, so the things they have observed, they appear large in size as compared to the original size. So that was the basic idea which Zacharias Janssen took, and then later on microscope was developed. So it was initially developed by Zacharias Janssen. Now we are moving towards the teaching points. Now later on, Leeuwenhoek made a much better microscope as compared to the initial one by the Zacharias Janssen and his sons, and he observed small organisms under it. Leeuwenhoek microscope has the magnification of 250x. This was comparatively better microscope, but not so much advanced, but this microscope is comparatively better than that of the Zacharias Janssen's microscope. Its magnification ability is 250x, means 250 times. It means that this microscope was able to enlarge the image of an object up to 250 times than the original. So this is a comparatively better microscope. Now there are two important terminologies we have to discuss here, and these two terminologies play a very important role in the microscopy field, and these two are basically based on the lenses. Remember, lenses is the base of any microscope. The advancement of lenses basically resulted in the development of various types of microscopes. So these two terminologies, these are: number one, magnification. Magnification is basically the ability of microscope to increase the apparent size of the image many times than its original size. whenever you use microscope, so people have observed, you students, have observed that there are lenses there. There are usually in compound microscope, in light microscope, there are usually two lenses. One is just above the sample, and one is inside the eyepiece. So these two lenses basically magnify the image of the object which is being viewed. Usually in labs, we prepare a slide and then we observe it under the microscope. So magnification is basically the ability of a microscope to increase the apparent size of an object many times than its original size. For example, in above example, the Leeuwenhoek microscope have the magnification of 250x. 250x means it can enlarge the apparent size of an object 250 times. So any microscope if have high magnification, so means it is comparatively advanced. Now another terminology we are going to study here, that is the resolution. Resolution is basically the ability of any microscope to differentiate two close things. Whenever you view various objects, particularly in biology, when we study the cells, so suppose you are studying the cell, so in the center of animal cell there will be nucleus, then there will be other organelles, endoplasmic reticulum will be there around the nucleus, and then further moving away from the nucleus, so there will be other organelles. You will find ribosomes there, you will find mitochondria there. So in biology and in other fields also, we need resolution. Resolution means we want to clearly differentiate the two objects which are very close to each other. We want to differentiate them from each other. For example, we are observing the human cell or animal cell, so we want to differentiate the nucleus from the nearby endoplasmic reticulum, which is found very close to the nucleus. So resolution is the ability of a microscope to show two objects separately. Separately mean to distinguish two things. When two things are very close to each other, so if a microscope have a good resolution, it means that it will clearly differentiate two very close objects. For example, human eye is just like a microscope, and it has its own resolution. So human eye can differentiate between 2 points at least 0.1 mm apart. If two objects are at least 0.1 millimeter apart from each other, and there should be at least 0.1 millimeter distance between two things, so the human eye will be able to differentiate these two things. It means that the resolution of human eye is 0.1 mm. So human eye can observe all those things which have distance of at least 0.1 mm or more. Through human eye, when we observe various things, two close things, if those two things which we are going to study, if these are at least 0.1 millimeter apart, so we can observe through normal human eye. Or if it is above than 0.1 mm, so we can observe those. But if the two viewing objects which we are going to view are close than 0.1 mm, so human eye cannot differentiate these two. Then the human eye will just recognize the two things which are very close as a one thing. So resolution is basically the ability of any microscope to differentiate two very close things from each other. And then you can recognize this thing is different thing and this is different. For example, nucleus and endoplasmic reticulum are very close to each other, but if we have a very good microscope through electron microscope, so we can observe that nucleus is different. We can recognize and identify the nucleus as different, and then the very close thing to the nucleus, that is endoplasmic reticulum, that will be identified as endoplasmic reticulum as a different item. So resolution is the ability of a microscope to differentiate among two very close objects. A very good microscope, an advanced microscope, will have very good resolution as well as magnification. For example, electron microscopes are the most advanced microscopes which have very high resolution and very high magnification power. Now we are going to study the light microscope. Now light microscope, why it is called light microscope? Because whenever we use microscope, so we need a light source to illuminate or specimen or sample. So light microscope is called light microscope because in this microscope the light source is the normal visible light, the normal light. Either sunlight or either artificial light inside the lab, or the small light which is present in the microscope. In both cases, these lights are called visible lights. These are visible lights. So light microscope are called light microscope because here the light source is normal light. This is called visible light. While on the other hand, in the electron microscope, the light source is electron beam. So that's why that is called electron microscope. And the normal other microscope which use the normal light, the visible light, this is called the visible light. So those microscope are called light microscope. So light microscope uses light to make the image of an object. Yes, we have discussed this. So light passes through the sample and then through two glass lenses. One lens produces an enlarged image of the sample and the second lens magnifies the image more. Now we have already discussed that in normal light microscope there are two lenses, one below just above the slide where your sample is, just above the sample, and another one is in the eyepiece, that is the second lens. So normally the first lens, which is just above the sample on the slide, in biology we make slides, so the first lens will magnify the image. Then when this light passes through the second lens, which is in the eyepiece, it will further magnify the image. So overall the magnification is so much increased. So the viewer's eye view the clear enlarged image. Its magnification is 1500 x 15 times and the resolution is 0.2 micrometer. Now the normal magnification of the light microscope is 1500 x, means 1500 times. The normal light microscope can enlarge the image of an object up to 1500 times, and resolution is 0.2 micrometer. Its resolution is 0.2 micrometer. It means that the light microscope can differentiate between two close objects when they are at least 0.2 micrometer apart. When the two objects are at least 0.2 micrometer apart, so they are clearly viewed through light microscope. But if they are very close to each other and their distance between the two objects is less than this 0.2 micrometer, then the light microscope cannot differentiate those two things. Then that will be just one image. Now we are moving towards some diagrams, some images. We will observe the microscope here in this image. Now look at the image. As we have already discussed, that this is the eyepiece through which we observe. We place our eye here and we observe. Then high power objective lens. This is the second lens, second lens which I have already discussed, that there are two lenses. So this is another lens, and here this is the stage, and on this stage here, at this area, here we will put our slide. So we put our slide here, then the first lens, this is the first lens, this lens, this is low power objective lens. This will magnify the image of this object on the slide, then it will be transmitted here. There in eyepiece there will be another lens which will further magnify the image, and as a result a highly magnified image is seen through this microscope. Through our eye basically observe the object, and the two lenses magnify the object. This is another diagram. Again, this is eyepiece. One lens will be here. This is objective lens. This is another lens. Here is the stage, and here will be the slide on which your sample is there. Again, the same which we have already discussed, that this first lens will magnify this image, then it will be transmitted above here to the eyepiece lens, and then here the viewer eye will observe the image. That's the end of today's lecture. We'll see you again inshallah with a new lecture. Take care. See you next time inshallah.